Good afternoon, everyone. Welcome back from lunch, and welcome to the Emerging Yale Biotech Showcase. My name is MaryAnn Melnick. I'm actually quite honored to be here in a different capacity, supporting and serving both Yale and New Haven. I live in New Haven. I've lived here for, I think, 13 years now. It's been going on for a while. And I used to work at the Yale Cancer Center, so a good part of my career has been spent in the Yale Cancer Center. So a lot of familiar names that I see on the panel or on the showcase here, and really excited to see you all spinning out companies from Yale. I'm the Director of Operations for Biolabs New Haven. Biolabs is a network of 14 co-working lab incubator spaces. We operate across the U.S. and we have several in Europe, in Paris and in Germany. What we offer is open as well as private, fully equipped lab spaces where a lot of the companies that we'll see in the showcase here can take full advantage of and focus their capital on their science and their R&D and not worry about the infrastructure and operational support. So I'm actually proud to say that about 25% of the companies presenting here are current Biolabs resident companies, and it's been thrilling to see them all grow in our space and become very successful. So hoping that more will come through, especially from the groups that are presenting here today. So I have the pleasure of moderating this session with Danforth Advisors. They'll do the second half. I have the first half of the showcase, and we're going to start off with Apira, Joseph Contessa.
Thank you for the opportunity to tell you about a company I recently founded called Apira. The mission of this company is to advance small molecule inhibitors of the oligosacchαρyltransferase for the treatment of cancer and other diseases. The oligosacchαρyltransferase is an enzyme that adds N-glycans to proteins as they're elongating in the endoplasmic reticulum. There are two partially redundant complexes that have two different catalytic subunits, STT3A or STT3B. Inhibition causes ER stress, and inhibition also impairs function of highly glycosylated proteins. So why have others not targeted N-glycosylation? The main reason is because there's a natural product called tunicamycin, which is toxic, and so there's therefore perceived toxicity about this approach. However, the targets in this pathway are unclear. Until recently, they have not been fully defined. The biologic effects of inhibiting these targets are not clear, and it's been very difficult to measure inhibition of this pathway. In addition, there are no small molecule inhibitors until what I'm about to show you. We did a high-throughput screen of over 350,000 compounds, moved on to chemical synthesis and structure-activity relationships to identify this first-in-class inhibitor, which we call NGI1. And you can see that unlike tunicamycin, which is on the end, we get partial inhibition. And this partial inhibition is quite important because it's the reason why we do not see much toxicity with our inhibitor. We went on to validate that there is partial inhibition using knockout models where we've knocked out either STT3 or SCT3B, and you can see in the red boxes that we get partial inhibition of both subunits. We also have found that we're inhibiting glycosylation of specific sites, which I don't have time to go into much more to tell you about, but it is a site-specific event. So the initial market is we anticipate will be sarcoma, and more specifically osteosarcoma. These are tumors that arise in the long bones and frequently metastasize. The global market for osteosarcoma is approximately $700 million per year. And more importantly, this is a disease with high unmet needs. And what do I mean by that? Well, chemotherapy doesn't work, radiation therapy doesn't work, targeted inhibitors and other agents don't work, and immunotherapy doesn't work. So it's also a rare disease that includes pediatric patients and has a very limited pipeline for drug development. So why sarcoma? We performed a screen of over 500 tumor cell lines to identify tumor types that are sensitive to OST inhibition. Our results shown in this whisker plot indicate that the tumors derived of bone or soft tissue were the most sensitive. And we also found that there are other tumor types such as lung and pancreas cancer that are also sensitive. So we've advanced drug development for this class of inhibitors in collaboration with New England Discovery Partners in Brantford, Connecticut, initially through an STTR grant, through private foundations, and now with Blavatnik funding. These efforts have advanced our potency by greater than 10-fold and our solubility by greater than 25-fold, which has allowed testing in preclinical models. And so the one I'm going to tell you about is NGI-155, which is circled here. So we use this in a xenograft of osteosarcoma, and we found that a relatively low dose of 10 mg per kg causes osteosarcoma xenograft regression. So this is an important finding because osteosarcomas do not typically regress in preclinical models. Our post-treatment pathology analysis of the xenografts show that we indeed are inducing ER stress, as you can see from the brown staining in the bottom here, as measured by induction of BIP, consistent with the mechanism of action. So is there toxicity? We looked at weight, behavior, blood counts, electrolytes, metabolic panels, and cytopathology, and we saw no difference between controls and NGI-155 treated animals. With these proof of principle studies and xenografts, we've now further advanced the inhibitor class, generating about 600 analogs for testing. These efforts have improved our solubility by at least 100-fold and our metabolic stability as well. And so that's our T1/2 is about 50 minutes in microsomes now. So our future goals are to identify leads with optimized in vitro ADME properties, perform in vivo PK studies, and then do our complementary in vivo tox and efficacy studies with the goal of filing an IND application. Thank you very much for your time.
Thank you, Joseph. Next is B3 Therapeutics. It's Mark Saltzman, but I believe Elias Cajano is stepping in.
All right. Thank you. Hi, everyone. My name is Elias Quijano. I'm a Blavatnik Fellow with Yale Ventures. And prior to joining this team, I spent the last 13 years really thinking about non-viral drug and gene delivery platforms. And what I've come to appreciate over time is that while many of them can be deployed for systemic disease, few, if any, really penetrate the central nervous system. At B3 Therapeutics, we're developing a novel polymeric system that's specifically designed for intrathecal administration, exposing the central nervous system directly to our therapeutics that are encapsulated. Thus far, we've shown that these polymer nanoparticles can encapsulate small molecule drugs as well as oligonucleotides. And so far, our in vivo proof of concept data supports the development of this asset for treatment of brain tumors. Our company has been founded by academic entrepreneurs. Mark Saltzman's, work in drug and gene delivery has gone on to form the basis of Stratify and Xanadu Bio, two Yale spinouts. Likewise, Ranjit Bindra, physician scientist at Yale, has gone on to found Cybrexa, Alphina, and Modify Bio. And of course, Kevin Rakin, known to many in the Yale community, has been a significant investor and co-founder of many of these Yale spinouts. Raman Baha has recently joined the team as well, bringing to bear tremendous expertise in synthetic chemistry. And lastly, I was able to join this team having recently founded Gennaro, which is advancing a monoclonal antibody for nucleic acid delivery, again, another Yale spinout. So when we think about brain tumors, it's obvious that there's a profound unmet need, be they diffuse pontine gliomas, medulloblastomas in children, glioblastoma in adults, or metastatic disease secondary to breast and lung cancer. What's common among all of these tumor types is that patients suffering from these conditions have poor prognosis as well as low survival. The primary barrier to effective therapy in these settings is the blood-brain barrier, which has been designed biologically to prevent the accumulation and passage of small molecule drugs. To overcome this BBB barrier, we and others have explored the idea of directly introducing drugs into the central nervous system. This has been done through convection-enhanced delivery. Here on the left, you can see an image where a radiolabeled tracer has been infused directly into the brain by CED. And what's obvious is that although the distribution is efficient within the first few hours, by 48 hours, the molecules have rapidly cleared. Likewise, intrathecal administration directly into the CSF has been explored. However, clearance of particles from this system happens on the order of hours. To overcome these limitations, we've gone ahead and advanced a polymeric nanoparticle platform, which not only rapidly distributes throughout the CNS following intrathecal administration, but has shown significant retention following IT administration. Here on the bottom, you can see fluorescently labeled nanoparticles that once administered IT rapidly distribute throughout the spinal cord and brain. And what's notable about these nanoparticles is that they're persistent. They actually last for up to 7 days following IT administration. When we take these spinal cords and section them, we can see that these nanoparticles rapidly associate with the meninges of the spinal cord and in many cases actually traverse into the central canal. On the right, you can see an example where we've now used PET imaging to trace the circulation of these nanoparticles once administered. And what you can see here in this model is that the particles rapidly accumulate within brain tumors. In this case, we're looking at a mouse model of medulloblastoma. Importantly, these particles are still detectable up to 21 days after a single IT dose. Using these polymeric nanoparticles, we've gone on to encapsulate DNA repair inhibitors. Specifically, we used for this example BMN 673, which is a PARP inhibitor. Here, using a mouse model of medulloblastoma. On the left, you can see we've introduced BMN 673 as a free drug, and you can see from the red line that no significant survival is appreciated beyond control. However, when this drug is encapsulated into our nanoparticles, you can see significant enhancement in survival as noted by the green survival line here up to day 100. We next sought to investigate whether or not this therapeutic approach was synergistic with temozolomide, the current standard of care. And what we found was that at day 100, this combination therapy actually resulted in 80% survival probability. While our lead indication and our lead product is really focused on developing polymeric nanoparticles to encapsulate and deliver DNA repair inhibitors, we have a number of exploratory programs that are ongoing, including the encapsulation and delivery of antisense oligonucleotides, DNA oligonucleotides, and plasmid DNA. As we continue to advance the technology and seek external support, we're really seeking to round out our set of in vivo proof of concept studies. And what we're primarily looking at doing this year is conducting non-human primate studies to really study the safety and translatability of this approach. Thank you.
Thank you very much, Elias. Next, we have Curatic's Joseph Gennaro.
Thank you. Hi, thank you everyone for joining. I'm Joe Gennaro, CTO of Curatix, which is a New Haven-based Yale spin-out out of the Choukri Mamoun lab here at Yale. We've assembled a team of scientists, entrepreneurs, and advisors, including doctors, and thank you, James and John from Yale Ventures for supporting us. Our mission is to develop more effective and safer antimicrobial therapies and advanced diagnostics. And we just got underway in April in biolabs on West Campus. We're supported by the NIH in our Phase 1 SBR grant. And antimicrobial resistance is an urgent global threat. In 2019, nearly 1.3 million deaths were attributable to it. By 2050, this number will grow to 10 million if we don't change our strategy. And we currently spend almost $14 billion a year on this threat. Now, this is a large, multifaceted problem, and we have a pipeline of projects, of two of which we'd love to share. They are pretty topical. You may have heard of both of them. Candida auris is a fungal pathogen, which was announced in March by the CDC to be an urgent threat because it's growing very quickly, almost 200% a year since 2016. And some of the cases are resistant to all known antifungals. So it's not yet Last of Us territory, but where are we headed with this? Babesiosis is a Lyme co-infection. It's a tick-borne disease. Like other in this category, 90% of cases go undiagnosed. In Connecticut, it's high on the list of growth since 2011. In some states, it's over 1,000%. And the CDC has noted for both of these, there's increasing incidence, ineffective treatments, and we need better diagnostics. So in the case of antifungals, we have the PAMS project, Potentiators of Antimicrobial Susceptibility. Now, the studies of fungal biology revealed if you destabilize the detoxification pathways against clinically used antifungal drugs, you potentiate their efficacy. So in vitro and in vivo, we've shown a protective effect for mice, and we consider this to have broad implications, not just for fungal infections, but also for antibacterials and antiparasitics. We're very excited about this project. And in the case of Babesia, studies into the proteins they secrete while hidden inside of red blood cells in your blood revealed a new form of diagnostic, which is very sensitive. It can detect in hemolysated or in whole blood at levels 10 to 20 times lower than what would be symptomatic. So this would enable a rapid diagnostic, a point-of-care test. You could use it as a prophylactic. Remember, the turning point of the COVID pandemic for our lives is when we could go visit, then get a at-home assay and determine we're not a threat to the people we care about. So we're very excited about this test, and we're launching it with the Yale Pathology Core in a CLIA-approved lab, and we anticipate setting up a service-based multiplex diagnostic here. And in both cases, these came out of basic research, but they've been vetted and they're getting funding in their own merits. And in the case of antifungals, we have a paper in Structure last year. This is high-resolution crystal structure of the target enzyme of the first class of PAMS compounds. And we hope to have, by the end of this year, $4 million in cumulative non-dilutive funding. And this is protected by Yale IP. In the case of Babesia, in April, we were on the cover of Nature. This is a life cycle of the parasite, little alien getting ready to bust out of the red blood cell. And here we're in Lyme Diagnostics, we're in negotiations for $2 million of a mix of dilutive and non-dilutive funding, again, protected by Yale IP. And this is just two of the projects we have. We consider ourselves a platform company. We want to take academic projects and de-risk them into commercial products, starting with non-dilutive funding. And we're looking for partners all along this value chain. And we think of this as a radical cure for the infectious disease pipeline. If you were at the VC panel earlier, you noted how quickly they were saying, well, this chain is broken. Even talent is leaving. We want to fix that. We think we have a way to do that here. So if you have any questions, please reach out to us, info@curetixbio.com. Thank you very much.
Thank you, Joe and Curetix, and thank you for the plug. We're very happy to have you as resident company. Intronex, Anna Pyle.
Well, I feel really lucky to follow the previous talk because this is also going to be about antifungals. Yeah, so you can't do more. It's just can't do enough, I should say. So thanks for coming to this presentation on intronics. So actually, can we look at this? I can't really see this on the screen. Because I want to be able to see you guys. Okay. Yeah. Thank you. Anyway, so thanks for coming to our presentation. We are interested in combating systemic fungal infections by targeting the Achilles heel of fungi, which is in their transcriptome, in their RNA molecules. Well, okay. If Mary says so, I better do it. So it's been founded by myself and Kevin Polizzi, and we have a wonderful set of business advisors and a terrific scientific advisory board. The overall company view is that our goal is sort of summarized in this picture here. We want to target RNA molecules that are unique to fungal organisms and do that with small molecules. And we are doing that using a patented high-throughput assay that was developed by people in my lab. And in this way, we are specifically targeting RNAs that are found in fungi and are not in mammals. And so we have a new solution for systemic fungal infections. We have broad expertise in RNA targeting and infectious disease, and our validated platform is going to be extensible to human-specific diseases as well. So we heard a little bit about this before, but the need for antifungal solutions is growing rapidly for a lot of reasons. for all of these important indications, but people may not realize a growing threat is the rise in infections that come from things that get implanted into the body. So implants, transplants are frequently complicated by systemic infections, and also immunocompromised individuals are particularly at risk. Fungal infections are really hard to kill, and the reason for that is that fungi are like us. They're eukaryotes. We share many proteins with them, we share a similar cell structure, but our RNAs are very different. But as a result, available antifungal drugs can be really toxic because they're going after those sort of traditional targets. And to meet this need, we've developed a set of compounds called intronistats that target RNA enzymes unique to fungi. So why is this important? It's important because existing drugs are not meeting the needs of patients. Many of them, as you can see, have been around for 50 or 20 years. They're many from the same compound class. They tend to be highly toxic, and even those that are well tolerated have to be taken IV. As a result, we just don't have many solutions in the clinic. There's very high mortality, growing resistance, as we heard about, and emerging untreatable pathogens that we read about in the news and actually now are watching on TV. So our strategy is to create new classes of small molecule antifungals that maximize efficacy and minimize toxicity. So how are we going to do this? We really want to target this unique class of molecules that are only found in fungi. So it turns out that many fungal genes, the mRNAs from those genes, are interrupted by little pieces of RNA. You can see the black line there. They have to be spliced out before the gene is functional. In fungi, that is accomplished by the fact that they form specific 3D structures that can be modulated and will respond to small molecule drugs if they're highly specific. So interestingly, those fungi of concern I'm showing you here on the left all contain one of the same molecular types that can be inhibited by similar molecular scaffolds. So what we do is that as a splicing pathway proceeds and the pieces of RNA are brought together, we have a sensitive molecular beacon that can actually catch it in the act and watch as it's inhibited by small molecules. And it does that by unfolding when splicing occurs and base pairing across the boundary of spliced genes. This is a very sensitive assay. It has many logs of sensitivity, and this enables us to screen huge compound libraries for effective inhibitors and also to do SAR and optimization. So this has already produced novel antifungal compounds. The version 1 compounds have been published, so I won't tell you too much about them. These target a very specific class of fungi. They're very potent, and they have an MIC that's comparable with the clinical standard of care, and they're active in vivo and bloodstream. What we're really excited about is our version 2.0 compounds, and I'm just showing you the inhibition curve for just a hit, something that's not even been optimized yet, so very potent, submicromolar. And then one thing that's really exciting is that in the lab now, we're solving the cryo-EM structures of the fungal targets with these small molecules to really jumpstart our SAR and to help us with optimization. So we're really moving quickly. And so we have started this company, so our business model is that we want to license existing assets that we have. We're going to use our lab to develop new ones and to extend our platform to new kinds of antimicrobial and mammalian targets. So right now what we're trying to do is raise our seed funding, hire our executive team, and transfer the knowledge to our corporate lab. I want to thank you and just close by saying if you're interested in RNA targeting and you want a very well-characterized target and system in which you want to get into that space, I think this is a good play. So thank you very much.
Thank you, Anna. Next, we have Luna Bioscience, Carla de Villers.
That's not mine. Actually, that's somebody else's.
Good afternoon. Luna Bioscience is pursuing an opportunity to capture a $1 billion veterinary vaccine market. We will displace multiple inferior products with our proprietary recombinant vaccine developed here at Yale by my co-founder, Dr. Joseph Annetz. Leptospirosis is a bacterial infection with annual global economic costs of more than $30 billion. It affects millions of farm animals, resulting in milk drop and the loss of offspring from spontaneous abortion. In the U.S. alone, economic loss from aborted calves may reach $2.2 billion a year. Dogs sicken and may need dialysis, and about 10% of infected dogs die. Leptospirosis is transmitted from animals to humans, causing serious illness and even death. Veterinary lepto vaccines are sold by the top 4 animal health companies and are based on mixtures of killed bacteria from a few of the 200-plus pathogenic serovars of Leptospira. Since the serovars differ by locale, regional vaccine customization is required. Luna Bio is developing a novel vaccine that will prevent all forms of leptospirosis using modern genomic analysis to identify conserved regions of the bacterial virulence factors. Our vaccine will offer a safe, universally effective, and affordable solution to protect against this devastating disease that will be superior to current vaccines. Our published results in mice and pre-publication work in hamsters demonstrate that our vaccine is safe and protects against leptospirosis. Animals were first injected with controls, or one of 2 different candidates, and then challenged with 1,000 times the lethal dose of a pathogenic Leptospira of a different serovar than present in the vaccines. Animals receiving the vaccines had no illness whatsoever, while all the control-treated animals died. As a directly relevant example of the competitive advantage of a recombinant versus killed vaccine, the recombinant Lyme vaccine for dogs rapidly captured market share from the established killed bacteria vaccines. It claimed 2/3 of the entire market within 3 years of its launch. The inventor of this recombinant Lyme vaccine for dogs is on our scientific advisory board. The market for a better vaccine against lepto is substantial and growing. Climate warming will increase the spread of leptospirosis globally. Within the next year, veterinary authorities are expected to designate Leptospira vaccine a core vaccine for dogs in all important markets. With hundreds of millions of animals in need of vaccination every year, the dog plus farm animal lepto vaccine market represents a multi-billion-dollar opportunity. We are being awarded a $600,000 NIH small business grant, which will fund final development of the recombinant proteins for the vaccine, i.e. materials sufficient for our first 18 months of trials. Top 5 global animal health companies, 3, have reached out to us expressing interest in conducting these necessary hamster and target species trials in a partnership. Discussions are ongoing. There is a low-risk, clear USDA approval pathway for animal vaccines so that our first product will come to market in roughly 3 years. We are a lean team with myself as CEO and Dr. Annetz as co-founder and senior scientific advisor. I come with operations and finance experience at Coca-Cola and Citicorp, and Dr. Annetz leads an infectious disease research lab here at Yale. Our SAB is full of heavy hitters with large networks. We will expand upon funding. We are seeking $4 million in funding to further develop and commercialize our new vaccine against lepto. With your investment, we will complete the required regulatory trials in hamsters and a pilot trial in dogs to enable rational planning of a pivotal trial in dogs. Future raises will be used to fund the pivotal trial in dogs and to begin pilot trials in cattle, horses, sheep, and pigs. The NIH grant is paying for antigen production, so our initial $4 million raise will be used to fund operations, to complete hamster trials, and to fund the dog pilot trial. We expect an additional $4 million and 1.5 years will be required to conduct the dog pivotal trial and subsequent USDA filing for the dog vaccine. Just to develop lepto vaccines for additional species, incremental smaller funding amounts will need to be raised. Thank you.
Thank you very much, Carla. Next company up is Panluminate on Samsad.
Hi, I'm Ansam Saad, and I'm co-founder and CEO of Panluminate. Today, I'd like to introduce you to a revolutionary new platform technology we call Panoptics. This is a technology which grew out of my PhD thesis here at Yale, and which we think will pave the way to true precision medicine. The fundamental premise of precision medicine is pretty simple. Take a sample from a patient, analyze it to identify critical biomarkers, and then develop a course of treatment that meets the precise needs of that individual. That, at least, is the dream. The reality, however, is a bit more complicated. It's been 20 years since the development of next-generation sequencing, and this dream still remains very distant. And so while everyone is still iterating on incremental improvements, we think the solution calls for something radically different, a solution that goes back to basics, back to the fundamentals of science. So at Panluminate, we have reimagined the entire process, and we innovated physically, chemically, informatically, and optically. And then we brought those innovations back together into a single integrated platform we call Panoptics. Panoptics is revolutionary because it's the only tool set that offers a complete solution to spatial biology. It is what we call progressive subcellular interrogation. This is kidney tissue. We start by physically expanding it slightly and then labeling it to identify cell neighborhoods. After that, this allows us to look more closely at individual cells, at single-cell resolution. However, we do this in 3 dimensions, unlike most single-cell spatial omics companies. But what is truly revolutionary and why we're very different is that we're the only tool where you can take the same piece of tissue and then go subcellular, and even down to the protein level at the resolution of electron microscopy, which has never been achieved before on a light microscope. And this is because we expand the tissue even more and we label it again, merging molecular and structural information to reveal cellular mechanics and function, which is the essence of biomarkers. 2 proprietary technologies enable Panoptics. The first is our ability to radically expand tissue samples 10,000 times in volume. And the second is game-changing labeling chemistry that allows us to do unlimited multiplexing in 3 dimensions and label not only proteins, but DNA, RNA, lipids, etc., without doing any damage to the structure. I focused on the kidney because it's our first priority market, but our KOLs have told us that there's a real need for this technology in brain pathologies and in cancer research, with the lead indication in breast cancer. So let me explain where we are as a company. When we met a year ago, we were still mostly in academic collaboration mode. Today, we are a fully commercial operation. We have over 20 paying customers. We have customers from pharmaceutical companies. We have customers from startups and from universities that are outside the Yale bubble. And In terms of technology, last year we had one foundational patent based mostly on my thesis. Today we have this revolutionary, groundbreaking multiplex technology that I talked about. And we also improved our core processing protocols internally, and we think that our processing experience itself represents a great competitive advantage. Operationally, last year I was on my own. I had great advisors, but it was mostly just myself. Today, we have 5 full-time employees, including new senior managers to oversee platform development and applications and optimizations. We formed a scientific advisory board, which includes specialists to advise us on polymer chemistry and labeling technologies. In short, we're a very different organization today. So where do we go from here? This is our development pathway. Essentially, in stage 1, we think about the stage as being data creation. So we're optimizing our protocols and learning from customers. Then we want to move straight into automation, creating a box to automate sample processing and imaging. And in the final stage, we actually want to monetize the data, make use of it, turn it into clinical and diagnostic tools to be used by people outside of our laboratory. So how do we take that first step? We are currently raising seed funding with a goal to raise $5 million to support us through 2025, and we would love to talk to any of you who may be interested in actually realizing the stream of precision medicine. And with that, thank you.
Thank you very much, Owens. And our last company for this block is PanV, Mike Cola. Scott White.
Here we go. Thank you very much. My name is Scott White, not Mike Cola, and I'm here to talk about PanV and our pan-antiviral antibody strategy targeting viral infections. First, I want to talk about the team. The original discoveries for PanV were made in the laboratory of Dr. Akiko Iwasaki, whose exceptional reputation and really I think the gravity of these findings have attracted what I see as a very serious development team that includes Mike Cola, who's got over 30 years of industry leadership, and his experience includes spinning out technology from several academic institutions, and scientific advisors that include myself, Drs. Cookland and Pierce, each of us with over 20 years of large pharma and biotech experience and expertise that ranges infectious diseases, oncology, immunology, and antibody development and production. I don't think I need to say anything. I think we all recognize our global vulnerability to viral infections and outbreaks, whether it's new viruses or newly emerging viruses. But what PanV's technology has to offer could really reshape the landscape in that it avoids targeting peptide epitopes, which are susceptible to viral mutation, and goes after what has been discovered as a carbohydrate moiety that's actually placed by the host cell protein synthesis machinery. The original discovery was made of a naturally occurring innate monoclonal antibody that binds to endogenous retroviruses and seems to play a role in preventing their reemergence. And you look more closely, what you find is that apparently the architecture of these envelope glycoproteins restricts glycosylation through the host cell machinery for protein synthesis and reveals a distinct epitope on the virus that isn't found on host cell proteins. And actually, if you go and look at other viruses that also have surface glycoproteins, you find that PanV1, this monoclonal antibody, recognizes these glycoproteins, whether they're in isolation or in the context of the intact viral particle, and again, from a wide variety of viral families. Exactly. this finding, if it can be brought through to a product, really has a chance to reshape the landscape for the treatment of viral infections through a single agent that could target a broad range of viruses in a way that depends on an antigen that's placed by the host glycosylation machinery. What this also does is open up several potential partnership opportunities based on interests in different types of infections, whether that be respiratory pathogens, vector-borne illnesses, emergence of reactivation of infections in immunosuppressed patients, or biothreat pathogens. And over the last year, with Blavatnik funding, we've been able to produce a humanized version of the antibody and better understand the nature of the original monoclonal binding to its target. What we're doing over the next several months is optimizing that antibody candidate to develop a series of lead and follow-on candidates that we would like to take into further development. We currently have a commitment of $5 million of equity investment from the Gates Foundation after having seen this information, and what we're seeking now is a co-lead for Series A funding to continue the development of the asset. And that's all we have. Thank you very much.
Thank you, Scott. How about another round of applause for the companies presenting in the first block? We have a bit of a break. The next block will start promptly at 2:30. So we'll see you back here in about less than 20 minutes.
That was the biggest thing, getting yeah, exactly. No one needs to know. Pictures, I suggest I left my car was Eichman, so I left my car.
I have one slide that is not in
It's also working.
Right, then the next one is gets out.
That's great.
Okay.
The lab's just right over there. Structures and
Can I work in your lab? Would take another 12 minutes.
Get structures, compounds balance. Usually these compounds can't visualize or spit out. It's fun to see that. It's so nice of you to come out and introduce yourself.
That was the solution. Let's do this.
That's what we're learning from our clinical trials.
Hi, Laura. So that's I'm not going to business school. What are you talking about? You're shaking my hat.
Nice to meet you. To bring in talent and have the students
You should have this back by now. 17 Yeah, tell me what they think of In fact, we have a visitor today.
Oh, wonderful. Oh, okay. Looking to kind of jump over this one. Yeah? Interested? Oh, no way. I see. I always say it like that at music pairs, you know what I mean? It's one of my friends that are saying, I'm like, that's I never saw that. Sounds like a good idea. Yeah. Some of you know, take your time.
Data sets is the big emoji answer. Yeah, no, I like that.
By the way, I keep saying what
Yeah, I've seen you. And you're
I meant to go for a second here before I talk to going through your own everything was there. Because I'm serving as an advisor for them. He's given us a lot of money by King and Tucker and iPhones. But I mentioned your company. I see. Would you be here for the neuro technique? This one is very advanced. We are discussing potential activity. It's a Yeah, because I try to help them establish one from scratch, and told them it's feasible, but it may be the best thing to work with an established CRO. You can just create a subsidiary. It's cheaper. The manpower is actually something to consider. If you're comfortable, so the conference is something as well. I think I'm going first to the Bloomberg conference. They're inviting me to speak at the market. I'm there from June 2nd to June 11th to that conference, and I go to Rabat for this conference on entrepreneurship. Let me know what you think when you want to the money to start, if you were to start like
because you know that one thing is to get the money, the other thing is to get
the king is offering them treatment by the water, so they are successful in getting more. So for companies like yours, you to bring a subsidiary there, but actually even with tax subsidies and yeah, it's growing fast, and because of the Abraham support, a lot of Israelis are coming back to Morocco, especially Jews coming back. So I actually know people about this in New Haven. Many of them want to invest in Morocco. So recently, a lot of future many have even told her that's a center.
We should just get up here together.
You need to visit. You will be contacts. When I visited in January, when they invited me to it's better than my the faculty.
Life is always one of those. Grab hands.
Thanks a lot. Thanks for inviting us. Thanks for what you do.
Yes.
Can you hear me? Hello, everybody. Okay, I think we're going to try to keep everything on track. Nobody's listening. Okay, let's go ahead and get started. We need a bell. So we need a bell. Okay. I'd like to welcome everybody to the second block on the showcase. My name is Bill Tanner. I am a managing director with Danforth Advisors. We're delighted to be a sponsor of the innovation event. We're also delighted to be part of the Connecticut biotech ecosystem. Just a real quick commercial on Danforth. Danforth Advisors, been around a little over 10 years. We're life science only focus. We do operational consulting. If you start a company and you need help with back office financial stuff, things of that nature, we provide consultants to do that. We've got a little over 200 consultants nationwide. We just started a practice in the UK. But our skills go all the way up to the C-suite. I'm a fractional CFO of several companies. We have everything in between. We have consulting as it relates to CDMO, CRO negotiations. We just started a CMO practice, Chief Medical Officer consultant practice. We do insurance and risk mitigation as well as strategic HR. So I think we're kind of soup to nuts as it relates to the operational support we can provide the companies. And what I always tell the companies that I work with, we'll take care of all the nitty-gritty, the dirty stuff. You just do you, because I know that a lot of the folks don't really want to do that stuff. So anyway, thank you everybody for coming back. The first presentation is Babbitt. It's Jaime Grutzendler. I hope you got that right.
It is my pleasure to introduce you to our Rabbit platform for precision therapeutics of brain and retina. My name is Jaime Grutzendler. I'm a professor of neurology and neuroscience. My partner is Roshan Gunasekara, who is the chemist behind this platform, and we work closely with Emmanuel Ashoka, who is a Blavatnik fellow. As you know, certain cell types in the brain and retina are preferentially affected in disease, yet therapies are rarely targeted to the cells most affected, potentially limiting drug bioavailability and efficacy and increasing the likelihood of side effects and toxicity. The possibility of targeting small molecules to selected cell types would improve treatment efficacy, tolerability, and would allow the use of drugs that would otherwise not be feasible for non-targeted delivery. As you know, endothelial cells line up the blood vessels throughout the body and are affected in many disorders. However, even centuries ago, it was recognized that the brain and retina vessels had unique properties that set them apart. However, we currently have no methods for selectively targeting the endothelial cells of brain and retina. Today, I would like to introduce you to our exciting Rabbit small molecules. Given their intrinsic fluorescence, we can visualize these small molecules and show that they enter the intracellular compartment, strongly labeling endothelial cells but no other cell types, as seen here on the left, following intravitreal injection in the eye, and on the right by live imaging of the mouse brain. We have a full understanding of the mode of action of these small molecules. Here, HEC293 cells were transfected with a mode of action protein fluorescently tagged to GFP. Notice that only the cells that express the GFP can readily transport the Rabbit molecules in red, despite the fact that there's many more cells there. The transport mechanism is conserved from mice to humans with a few distinctive additional cells expressing it, in particular the retinal pigmented epithelium and oligodendrocytes, which opens up important applications for retina and neurological disorders. Here, I'm showing you a proof of concept demonstration of our ability to conjugate a rabbit molecule to a well-known drug like colchicine, which is a microtubule inhibitor with anti-inflammatory properties. On the right, I'm also showing you evidence of the preservation of the potency of the rabbit colchicine conjugate compound. Importantly, these conjugates retain their original cellular specificity as seen here in the live mouse brain. Strikingly, we have been able to conjugate and deliver large hydrophilic compounds of molecular weight greater than 2 kilodaltons that would otherwise be nearly impossible to deliver intracellularly and selectively. And here is a dramatic demonstration of the complete elimination of colchicine side effects. In this experiment, we shaved the fur of both mice. We administered daily subcutaneous injections of either colchicine on the left or rabbit colchicine conjugate on the right. Notice that the fur regrew perfectly on the right, but not on the left. This demonstrates that skin cells and follicles which do not express the transport mechanism are completely spared from the effect of the rabbit conjugates, thus eliminating the prominent fur growth toxicity of colchicine. Given the affinity of rabbit compounds for endothelium as well as retina, pigmented epithelium, and oligodendrocytes, we have identified a number of potential disease targets, including age-related macular degeneration and neurodegenerative diseases affecting vasculature and oligodendrocytes. As an initial proof of concept, we're focusing on age-related macular degeneration, given its well-understood mechanisms, availability of animal models, and proven regulatory path to approval. We plan to target both the angiogenic vessel overgrowth component of AMD as well as the inflammatory and degenerative component affecting the retina pigmented epithelium. We aim to design a conjugate that will be orally bioavailable and that we believe will be synergistic with current available anti-VEGF antibodies that require repeated intravitreal injections. We have identified a family of JAK-STAT inhibitor compounds with anti-angiogenic and anti-inflammatory properties that we believe will address both of these mechanisms of AMD. We believe that this will synergize with current therapies for AMD and will resolve issues with poor long-term compliance and effectiveness of current intravitreal injection approaches. We are looking forward to connecting with prospective collaborators and investors to advance our platform and explore additional applications. So together, let's build the rabbit platform for precision therapeutics. Thank you for your attention.
Thank you, Jaime. Our next speaker is Greg Tietjen from Revalia Bio. Very excited to introduce Greg. Danforth started working with the company in the very early days. Hopefully we've been helping them. And we still do. So, Greg, thank you.
Yes, thank you, Bill. We can definitely be a poster child for biolabs, for Danforth, for Yale Ventures. We're a Yale spin-out company, so all have been fantastic and instrumental in the progress that we've had to date. We're a pre-seed company, and I was formerly a Yale faculty member up until about 8 weeks ago. Took the full leap in without much of a safety net, but because I believe deeply in what it is that we're trying to accomplish at Revalia. Perfect. And what it is that we're trying to accomplish is really a new play on trying to transform the process of how we do biomedical innovation. We've heard about so many amazing technologies here today and yesterday too, but there's so many challenges in the process as it exists now. And in particular, we'd like to summarize it by saying this system as it exists doesn't fail very well, right? So we all know this pipeline. The problem is it takes an incredibly long time to fail in the setting that we most care about, which is a human with a disease of interest, if it's a drug, for example. And the consequences of that failure are massive, right? Obviously, we put patients' lives at risk, but there's massive economic consequences. It can reach $1 billion, right, depending on the stage of the development. And the failure isn't useful in the beginning stages when we could use failure because we often start with the wrong systems. We don't begin with the patient, with the end in mind, with a highly variable patient population. We begin with what's available to us. So at Revalia and what we've done in my lab for the last 10 years is come up with a new thesis, which that if we can recenter on a new system, which is donated human organs that are not usable for transplantation, but can be used in research settings, then we can transform this process. Because first and foremost, we begin with the end in mind. We start with a highly variable human population. We can go incredibly fast, so we can actually sort of start with the clinical trial in the beginning, so we can be very agile about it. And the whole thing is we're not putting patients' lives at risk, so we can actually, it's allowable to fail, and we can transform the catastrophic failure in a clinical setting into a catalytic aortic failure in this setting. And while doing so, we can actually meet the needs of donors' families who also suffer a catastrophic loss when they're told, I'm sorry, your loved one's organs weren't good enough for transplant. And the reality is that for a patient that passes away in ideal conditions for donation, over 90% of the time, that's what those families are told, because that person has cancer or other things that are rule outs for transplant. But they often desperately want to have something meaningful happen afterwards. If your loved one loses from cancer, you want to continue the fight and help in some way possible. So what we're enabling is the ability to receive those organs at any time of day or night, just like we would be able to do in a transplant setting, bring them back to life, and prove that an organ doesn't have to be transplantable to be transformative. So what does that look like? Well, it starts with these systems. So what you're looking at is a machine perfusion that's actually used clinically in the transplant setting. The way I want you to think about it is a mechanical patient, right? So this mechanical patient, it actually has heart, lungs, we've modified to have kidney, we control nutrition, and it has a brain that acts as an algorithm so that when we bring this organ back to life, unfortunately the video's not playing, I'll show it to you all later if you want to see it, but what you would see there is the liver being reperfused, coming back to life, and when it does, it'll do things that livers do. So it'll synthesize proteins, it'll detoxify, it'll also propagate the underlying injury associated with the disease, which in this case is sterile inflammation associated with fatty liver disease. We've developed with some partners the ability to go out to 5 days so that you can study that dynamically and evaluate not just disease pathophysiology, but the ability to test interventions. And we do that by leveraging our real expertise, which is the ability to sort of speak the language of the organ and extract all of the knowledge using all of the cool tools that we have, from 3D rendering of organs to quantitative histology, multi-omic technologies, and even could be a breeding ground for all the amazing new spatial omics and technologies we've heard about here today. The really cool thing is that you can now functionally analyze the organ as well. So this is an example of some work we did in a science paper several years ago, testing the ability to improve bowel quality in an individual organ. So what we do is really a partnership place. We are a catalyst company that is seeking to really accelerate and de-risk high-quality innovations with partners. What our expertise is around is being able to design these high-complexity experiments, execute them, and deliver high-quality data. And we've got a full-stack technology team that is behind this, building all this out. This is an MVP version that started at Yale, but we now have a full tech team building out, or actually have released our second version. And with that, what we're able to do is really fill a gap between animal and human clinical trials, and leverage the inherent complexity and variability of human systems, which is the point. We need that. That's what our patients look like, so we need that to be able to develop the appropriate therapies. We can be forced to think about some of the late-stage problems like delivery and other scale-up bottlenecks that, if we wait until the end, we can't actually scale that up, or we can't actually deliver it effectively into a patient. So that's why this becomes a catalyst. And I've sort of talked about in the context of therapeutics, and that's where we started for, but it really isn't limited to that. It really can apply to different, diagnostic tools or medtech devices, and we're really exploring across the landscape. The challenge is being able to scale it up and deliver really on the speed. And so that is what led us to leave Yale, because it wasn't possible to really solve those problems in the academic setting. And so we've built now a full stack development team. It's a flexible staffing model. So this is, I'll finish fast. This is our 6 team. We have 10 teams behind it building new software and hardware. We have an amazing perfusion team that's breaking new ground, including recently doing the world's first triple liver perfusion for research simultaneously. And we've built out a bunch of relationships to build a network out to do this. We now can work in lung, kidney, and liver, but have ability to expand into heart and pancreas, expanding into cancer, multiple different style of perfusions that we can do, and multiple different types of injury and disease models. We have an incredible team that we've assembled, not just our executive team, but advisors who are really guiding the way on making not just very exciting science and technology, but really a very scalable business that can be around to make sure that we can drive as many innovations into patients as we possibly can. Thank you very much.
Next presenter is Ray Johnson with Saga Bio.
Sure. And what's my... All right. So, glad to be here. I'm Ray Johnson. I'm currently have all those titles for Saga Bio Incorporated. That's temporary. We're a translational immunology and immunodiagnostics company. Our first project is Kawasaki disease. That's a febrile vasculitis in children that affects coronary arteries. Typically, the children are between 6 months and 6 years of age. The diagnosis is based on a fever of 5 days plus 4 or 5 possible physical findings. To medical eyeballs, Kawasaki disease looks like a viral illness, and it's easily missed by pediatricians and family practice physicians. Angiographic studies suggest that 2/3 of cases in the United States go undiagnosed. The treatment for Kawasaki disease is to hospitalize the child and deliver pooled human gamma globulin. That's essentially antibodies. So there's a need for a rapid diagnostic test for Kawasaki disease to guide what is ultimately an expensive therapeutic decision. So here's our science. Do I have a pointer? I don't know, do I have a pointer? But anyway, you can see in yellow there that while Kawasaki disease looks like a virus to medical eyeballs, at the cytokine level, it looks like a severe bacterial infection with, I have to look backwards now. It looks like a severe bacterial infection with very high levels of soluble TNF receptor 2, which is a readout for TNF alpha biology. And not surprisingly, Kawasaki kids also have elevations in vascular chemokines because you have blood vessel inflammation. So this is the algorithm from our published paper. Basically, this works because viral infections are about interferon, bacterial infections and Kawasaki disease are about TNF alpha, and Kawasaki disease includes blood vessel inflammation. So if you're a febrile child, your soluble TNF receptor 2 level is low, you probably have a viral infection and you can go home. If it's markedly elevated, that means you're very sick. If it's markedly elevated and your vascular chemokines are also elevated, you have Kawasaki disease. So it's all biomechanism based. So we're developing a soluble TNF-R2 point-of-care test. That's more than a Kawasaki disease screening test. It turns out soluble TNF-R2 is a really good biomarker for serious inflammatory events that probably require hospitalization. You can use that to simplify febrile child evaluations in emergency rooms. It also identifies children who might have Kawasaki disease for additional evaluation. The market's quite large. Fever is the #1 complaint in pediatric emergency rooms and 30% of outpatient pediatric visits. We're going to revisit that figure down in the corner in a coming slide. So the technology barrier was no antibody pairs for capturing and detecting soluble TNF-R2, so we've made those monoclonals. We currently have a CLIA certifiable soluble TNF-R2 ELISA that performs better than R&D system kits, and we have a soluble TNF-R2 lateral flow point of care prototype device, which is shown there, and its performance characteristics are just to the right. So our relevant competitor is procalcitonin. Procalcitonin is a really lousy screening test for Kawasaki disease because in children with Kawasaki disease, the level of procalcitonin is not statistically different than children who have viral disease. So that's what you're trying to distinguish. Whereas if you look at soluble TNF receptor 2, it's very high in Kawasaki disease, KD. It's still high in serious bacterial infections. It's still elevated in severe viral infections that require hospitalization. And then it's low in healthy children. That's kind of the pattern you want. It also matches up your risk. So our revenue stream, the soluble TNF receptor 2 ELISA is pretty much ready to go, as is. The next would be the soluble TNF-R2 point of care test. We have a pipeline. We have therapeutic anti-inflammatory monoclonal antibodies that have already been humanized and are in preclinical development. We have the intellectual property to support all this. Our lead academic investigator is Kelly Bergman. He's a director of emergency medicine research at Children's Minnesota Hospitals in in Minneapolis. Our lateral flow device development partners, Nanocomposics. Our funding is going to come through a phase 2 STTR. And as of today, we are a physical company as we've rented space at Biolabs New Haven.
Thank you, Ray. And one thing they wanted us to mention is these presentations are all saved. I guess it's on the website. So Ray kind of went through the last part, but you can go back and look at his presentation or slide deck. So the next speaker is Angelique Bordey from Neurolexa Therapeutics. Angelique, thank you.
Hello, can you hear me, actually? Yep. Okay, very good. Good acoustic. I think it's okay, right? Okay, that's good. Thanks. Well, we will switch gear a little bit because we have been interested in epilepsy and treatment for epilepsy in an orphan disorder called tuberculosis, TSC. Switch slide. Is that working? Or should I move it here? Last 10 seconds. So anyway, the next slide will show you why TSC, what is important about TSC. So this is an orphan disorder and the patients have genetic mutations, and the most dramatic phenotypes of these diseases is actually epilepsy. 90% of them have severe seizures. And it might not work. It works. Thank you. Yeah, I'll do reason. So 90% of them have seizures. They're diagnosed around a medium of 3 months of age. They will have seizures for life. And we actually know the cause of seizures. This mutation occurs during fetal development in brain cells and lead to this kind of brain malformations. One of the standard of care in 10 to 15% of the patient is to remove these brain malformations. The other standard of care is the drug sirolimus, but both of them don't work very well and have terrible side effects. So we really need better treatment for this patient. On top of it, there are a lot of comorbidities with seizures that includes insomnia, learning disability, and really behavioral problems like anxiety. So there is really a cry by the patients for better treatment. This is, although it's an orphan disease, there are about 30 to 40,000 people who have anti-epileptic drug-resistant epilepsy in the U.S. The cost is actually quite enormous, $16,000 per patient per month, which gives you a market opportunity of $4 to $5 million a year. So I've been working on TSC for the past 12 years and have very close ties with the TS Alliance. I'm on the advisory board for the preclinical consortium and look at their leads, clinical leads. And we can have a direct access to quite a few of the patients. The TS Alliance oversees 68 TSC clinics across the U.S. And then competitions, well, we're not the only one to realize that we will find a treatment in 5 to 10 years for TSC epilepsy. And there was a new kid on the block, Epidiolex, that was approved in 2020. That drug does not work well. 20% seizure reduction, terrible side effect. We may have another drug, the MEG blocker, but as you know, that drug has also terrible side effect and actually came from our own work. So that's why it's just complicated. And I'm sorry that I don't have the animation. I forgot to put it in. But basically, if you focus on this thing, they have mutation that leads to increase in mTOR activity. It's a really important intracellular hub. That's the target of the standard of care, everolimus. What mTOR does, he makes big cells. That leads to seizure. So really, what I want to show you in a minute is why do they get seizure? What does that mean? So here's a normal cell. It's a little small here. This is a neuron in the mouse cortex. We have them. They're beautiful. They are processes. They receive and they make connections. It's a normal one. In the disease condition, that's the one. Here it is. Very clear. The cell is bigger. mTOR does that. Because there's an increase in protein synthesis, they acquire the wrong channels and they're hyperexcitable. One of these channels is HCN4, which makes them pacemaker like your heart. The other thing that is obvious is connectivity. We're all making connectivity in our network today, but in this case, this is not good. It's over-connected, and it goes all over these cells, sun projection to all over the brain, and trigger or make a seizure travel farther. So what we did is that we blocked these increase in protein synthesis by overexpressing that little molecule 4-ABP. We turned that cell back into that one, and that reduced seizure activity. And this is a target for gene therapy. Local injection with neurosurgeons, since we have local brain malformation. The other things that we found accidentally is the overexpression of that molecule, filamin A. So filamin A is actually acting like a VC. It has a lot of binding partners inside the cells, and it allows the wrong partners to be expressed in that cells, which triggers excitability. When we blocked filamin A function or knock it down, we turn it back to that, and that blocks seizures. So that's what we did. And the work in mice is translatable to human because the same pathology, and we monitor seizure activity. And we're at the stage of seed where we need really money to make new drugs that are better and that we will own, and having a composition of matter patent. And we would actually also like to have, I guess I lost my slide, the last slides. And we also would like to eventually try gene therapy, which takes more time. Thank you for your attention.
Thank you, Angelique. Next is Ashoka Madduri with Target Site.
Excellent. Good afternoon. Can everybody hear me? Yes. Excellent. Awesome. Ashoka Madduri. I'm a Blavatnik fellow at Yale Ventures, and also joined Target Site Therapeutics as their CEO last year. We are basically here harnessing the power of novel mRNA targeted oligonucleotide therapeutics. So basically, company formed in 2019. So since the inception of the company, we are fortunate to receive 2 Blavatnik prestigious grants, along with SBR grants and also internal Yale grants to support our research. In a nutshell, we are building an mRNA targeted therapeutic platform company focused on unique insights into mRNA stabilizing microRNAs. I will get into it in a minute. So we have a lead candidate. It's also partially de-risked in the 3 disease models, one in MS, psoriasis, and autoimmune uveitis. And also, we recently formed a collaboration with a public company. It's optimizing oral delivery formulation to target immune cells to deliver our oligonucleotides orally. Orally is the bonus, actually. I think our focus more is on the target immune cells there. We have a strong IP from Yale. We're looking to raise capital and also build partnerships. So in a nutshell, our technology, the key differentiation factor, as you know, there are plenty of oligonucleotide companies out there. So the way we are focusing here on the left-hand side, on the right-hand side I want to start, then I'll come back to the left-hand side. This is a key story for us. On the right-hand side, most of the microRNAs are repressive in nature. So what that means is, so when microRNAs bind to 3' UTR of mRNA, they destabilize target mRNA. So what we are doing is we are developing target site blocker that destabilize target mRNA by using microRNA technologies, by enhancing microRNAs. So this technology developed from Jeffrey Bender lab. So what they did is actually they discovered enhancing microRNAs. What they do is actually they do exactly the opposite function of repressive microRNAs. When they bind to enhancing microRNAs, they stabilize the target mRNA. So what we are doing here is on the left-hand side, we designed the target site blocker. Target site blocker is a oligonucleotide anywhere from 15 to 23 nucleotides that takes the place of enhancing microRNA, destabilize the target mRNA. So for diseases like autoimmune and inflammatory diseases. We do have some data also stabilizing target mRNAs, but I will focus mostly on the left-hand side. So here is to show the way we design our oligonucleotides, how they show the specificity. As you can see, this is the in vitro data from human T cells. Both IL-17 mRNA and GM-CSF mRNA have same microRNA target, MIR466. So despite the fact that both mRNA transcripts have same microRNA, the way we design our target site blocker, it only destabilizes the IL-17 mRNA on the left-hand side compared to GM-CSF. So CNTA is the control oligo. As you can see from left IL-17 to GM-CSF, there is drastic degrees of IL-17 mRNA compared to GM-CSF. So we do have plenty of in vivo data in 3 disease models. Here I'm showing one of them because of the time. So it's one of the progressive MS model data in vivo. As you can see, we injected our target site blocker at day 6 toward the disease course, every 3 days toward the disease course. As you can see from the graph here, at day 21, the EAE score is 5, means almost all mice have died. So as you can see from the control oligo CNTA at day 21, all mice are died. With our TS target site blocker, all mice are alive and kicking actually. So this is really a impressive data that we got for one of the MS models that we have. So the near-term plan for us is, as I said, we are working with one of the delivery companies. It's a company called Madduri Bio. They develop lipid nanocrystals. By using that platform, they can deliver small molecules and oligos orally. They do have phase 2 data in small molecules, and now they're applying their technology to our oligos to target immune cells. That is our main focus. Then bonus is actually oral, actually, if you can make it so. That's what we're doing now in collaboration with Madduri. Currently, we are conducting experiments. In a few months' time, we should be able to get the data to progress one of our lead assets into IND-enabling stage. So disease focus currently MS, and also we are looking into IPF. So this is our team. And most of the work, as I said, came from Jeffrey Bender's lab. He has appointment in cardiology and also immunology, and he is our SAB chair. Most of the work presented here and what currently done by Vinod Ramagolam. He's a student scientist at Jeffrey Bender's lab. And we have support from Yale Ventures for BD. If there are any questions, happy to take. Thank you.
No, but I didn't find a black hole.
We ready? Okay, Choukri bin Mamoun from Virtus Therapeutics is the next presenter.
Thanks everyone, and thanks for the opportunity to present today. This company also spin-off from my lab. Virtus Therapeutics. Pointer is not working. And I am the founder of Virtus Therapeutics, and our primary mission is to develop the first treatment for pentatonide kinase-associated neurodegeneration, or PCAN. This is a terrible disease. It's a Alzheimer of children and young adults. It results in Parkinsonism, dementia, inability to control muscle function, and death. And it's caused by a single mutation in the pentatonide kinase 2 gene, which is located on chromosome 20. I was invited last week to speak to the parents of many children, and I can tell you it broke my heart. This is really urgent and important. See, and that's many, many of these children. Lucy, for example, right here, was diagnosed only 3 months ago. She was a normal child, played soccer, doing everything else. That child, in a few months, will be in a wheelchair, and don't know what will happen in a few years. Just an absolutely devastating disease. The disease can be diagnosed often 3 years after it happened by MRI due to the accumulation of iron in the basal ganglia in the brain. From a basic biology standpoint, this is related to the metabolism of vitamin B5. Vitamin B5 needs to be phosphorylated into phosphopentatoate, which serves as precursor for coenzyme A, and coenzyme A is a major cofactor in cellular metabolism. Humans have 3 pentatonite kinases. 1 and 3 are cytoplasmic enzymes, and the PANK2, which is mutated in patients with PCAN, is a mitochondrial enzyme. So the problem is when PANK2 is missing, you have an altered CoA pathway that results in altered mitochondria, neuron, and brain. The solution we are offering is a class of compounds that were discovered in the lab that are activators of the second enzyme, PANK3, to compensate for the loss of PANK2. In this case, you restore the CoA pathway, mitochondrial function, neuronal function, and brain function. This was discovered from a lot of work that was initially sponsored by Program Therapeutics for Connecticut Health from a library of 156,000 compounds, modulators of pentatonite kinases. We identified 14 compounds that activate human pentatonite kinase 3 in the nanomolar range. We received the Blavatnik Award, and we are expecting funding, non-dilutive funding, in July of this year, about $700,000, to continue our cell-based and animal studies. And we will be, by the end of this year, early next year, starting our Series A raise to enter phase 1, phase 2 clinical trials. We know lots about this disease that I don't have time to speak about. For example, we know that they act through 2 mechanisms of action, active site activators and allosteric activators. And more importantly, the only other compound currently in clinical trial is by BridgeBio, which is basically an inhibitor of human pentatonite kinase 3, but serves as an orthosteric activator. It activates in the presence of acetyl-CoA, and as you can see, in our hand, it does nothing. Ours are direct activators of the enzyme, so we really have no competition in this case. The compound has a very long half-life, especially VPAC2, which can be administered once a week. It has CNS exposure 2 to 3 times their AC50 in the brain. We developed the first cell-based assay to screen for these compounds, where we can determine cells that are normal with PANK2 have a luciferase activity, cells that are missing don't have luciferase activity. That allows us to conduct detailed SAR. And these molecules were produced, I don't know what you don't see here, but it's actually mice that we produced that like PANK2, and we've already administered our compounds to these mice, and the results are being collected. We have a second mouse model also that we are developing. The breeding is taking forever, but in 9 months we'll be able to deliver those drugs to also that second mouse, and we hope to get all the data so that we can start phase 1 clinical trials. So in summary, a novel set of activators that act through novel mode of action, we have a novel strategy, and we have an IP less than 2040. We'll be raising Series A, so we are looking for those partners who are interested in helping us fight this disease. Our goal is to enter IND in phase 1, potentially even phase 2, and our goal is to develop the first treatment for PCAN so that we don't have to see this. This is what I was invited to see. These children that die, and their families are absolutely broken. And these are all the families that you see here, and this is what they have to live with. So really something that we can do something about. It's a rare disease. We are not getting traction from large pharma, but we have something that we can do about it. So hopefully, if it cannot be done with money from VCs, we are going to do it through non-dilutive funding, and that's what we are aiming to do. Thank you very much.
Thank you, Choukri. Next speaker is Chen Yan from Methyl-Zyme.
Hello, good afternoon, everyone. Let's see.
Okay, so I'm happy to present to you Mesozyme, which is it's dedicated to develop epigenetic immunotherapy. So as all you know that immune checkpoint blockade and other immunotherapy have achieved great success. However, they do not work on some of the tumors, some of them, especially some of the resistant tumors. So what do? We found a new target called Cadin-5B. We have shown that in the immune checkpoint blockade resistant tumor model, YAM 1.7 melanoma models, if we get rid of Cadin-5B, you can have some effect. You can combine with PD-1 blockade, you can double the survival of those animals. In a more similar model to patients, where it's called YAMA 1.7, where the melanomas has been exposed to radiation so that have more antigen and mimic the patient. So you can see in this model, when you get rid of Cadin-5B, the tumor initially can grow, then they got fully rejected. Moreover, if you re-challenge those animals with the control cells, which normally can grow well, they never grow up. So this suggests there's a strong memory response, and it can prevent the recurrence of those tumors. So we have solved the mechanism, and we showed that Cadin-5B can silence the retroanimal to promote immunotherapy, immune evasion. So however, Cadin-5 inhibitors do not work in this setting, even though we spent many years to develop Cadin-5 demesase inhibitor. So our solution is to develop Cadin-5 protein degraders, so which many of you know this very well, basically utilize the ubiquitin system to target the target protein for degradation. We have developed a platform to screen for Cadin-5 B degraders. Basically, we use the reported cell lines where we have the Hibitag Cadin-5B, and once it's expressed with large beta, you can have the nanomer surface. We have identified hits that can degrade Cadin-5B specifically, or can degrade both Cadin-5B and Cadin-5A. Cadin-5A, although I don't have time to mention, is also a very important oncogenic target. So this is our team. I have been working in this field for many years, and since this discovery, our team, Dr. Chang here, has developed the screening platform. Our collaborator, Dr. Jin, is a very experienced medicinal chemist who have advanced multiple drugs in the clinical trials, including one has been approved. So what do we have here? So we have novel discovery, we have the platform for screening, we have no first-in-class Cadin-5 degraders, and we have unique models to test those drugs. And we have already have partnership interest from pharma and venture. We actually set up a collaboration with some of the pharma as well, and we're supported by Marketing Fund, and we still have another year to go, and we are looking for Series A funding to advance this to IND filing. I went a little bit faster than others. Thank you all.
Thank you, Chen. So the next speaker is Ryan Muldoon from RNA Connect. Thanks, Ryan.
You're almost done. 2 speakers left. You're going to make it. So I'm Ryan Muldoon. I'm co-founder and CEO of RNA Connect, and I'm really grateful to have the opportunity to introduce the company to you today. We founded RNA Connect 3 months ago with a mission to provide better tools for RNA researchers. And specifically, what we have are a suite of enzymes that are foundational technologies for interrogating RNA. We think the existing suite of enzymes that everyone uses are, in a word, terrible. And so we're working to provide better ones for the community. We think what that will enable is whole new levels of innovation, because when you have better tools, you're able to create bigger, better, faster things. So for us, our lead compound, our lead enzyme, is a novel and highly differentiated reverse transcriptase. We think what this will allow us to do is have dramatically better viral pandemic surveillance. It'll enable us to do better target identification and biomarkers for cancer and neurodegenerative diseases. And importantly, it's going to provide dramatically better applications in RNA sequencing. So what is reverse transcriptase? It is an enzyme that latches onto single-stranded RNA, and like a zipper, it creates a complementary strand of DNA, which can then be amplified for your PCR COVID tests. It can be sequenced by the DNA sequencing machines from Illumina and PacBio and others. And there are many reverse transcriptases that are on the market already. The trouble with them is that they are from the early 1970s and were in no way designed to do what we're using them for today. So we have all sorts of workarounds to overcome their shortcomings, which are specifically related to processivity. What that means is it's how long the enzyme will continue copying the RNA template before it falls off. And the existing RTs will go 100 to 200 bases before they simply fall off the template. That's a problem because most RNAs are 2,000 to 30,000 bases long. So you end up with these little snippets of information, but you're never able to see the whole RNA. This is confounded also by the fact that RNAs are highly structured. They have all sorts of loops and stems and secondary folds in them, and each time the existing RTs encounter one of those, again, they fall off the template. So there's all sorts of information in those secondary structures that are essentially invisible to us right now because the RTs can't get through them. So what is our solution? Our solution is Marathon RT, Marathon Reverse Transcriptase, which is a novel RT that was discovered right here at the Yale Pyle lab. It was optimized and the structure was solved here. And when it was initially tested, we looked at, okay, well, how processive is it? Can it read through an RNA transcript? And we gave it the Hep C genome, 10,000 bases long, and it read the entire thing no problem. This was fairly astounding when we said, okay, well, what else can it do? And I love the expression for anyone that's at least my age. We gave it what Dr. Pyle likes to refer to as stupid pet tricks. And we inserted all sorts of little RNA structures, secondary structures into it and said, okay, well, can it read through? And let's compare it to the market-leading reverse transcriptase, which is superscript 4. And what we saw was superscript 4 does what it does. Every time it hit one of these inserted structures, it fell off the template. But Marathon RT read all the way through. It unwound all of those structures and gave us the full cDNAs for those RNA transcripts. So this is a vastly improved reverse transcriptase that will enable all sorts of new applications, particularly related to RNA sequencing. Imagine now we can get better single-cell sequencing. We can read all of the RNAs within a single cell. So what we're doing is we're packaging this enzyme. It's already fully optimized. It's ready to go. We're putting it with the other reagents into kits, and in the next 6 months, we'll begin commercializing them. So this will be for all sorts of sequencing applications, as well as for RT-PCR applications. We'll have products in the market in the coming months. Ultimately, this will enable new diagnostic applications, companion diagnostics for cancer, for neurodegenerative diseases. So we'll be looking to start those programs as soon as we can get the company off and running. The company was founded, and we have a scientific advisory board that are RNA researchers. These are leaders in the field, people that many of you, I'm sure, know, that are helping us then shape, okay, so what are we going to do with this enzyme as well as the rest of the enzymes that are in the portfolio to come. So I thank you for your time, and we look forward to enabling all of those that are in the RNA research community with our new tools.
Thanks very much, Ryan. Saving the best for last, D2B3, Ann Eichman. Thanks, Ann.
Okay, good afternoon, everybody. I'm very excited to be here. Okay, so very excited to be here. My name is Ann Eichman. I'm a professor at Yale and Innovator Awardee 2022, and I'm here to pitch D²B³, which is a new startup that enables drug delivery, D², across the blood-brain barrier, B³. So together with my team, composed of scientists and a businesswoman here at Yale, we developed and patented monoclonal antibodies that transiently open the blood-brain barrier. So you know that most drugs do not cross the blood-brain barrier, and therefore most brain diseases cannot be cured, and this is a major unmet medical need. Only 2% of all small molecules cross the blood-brain barrier and can be developed as drugs, as you heard earlier today already. The vast majority of all other molecules do not cross and are not developed as drugs. And so D²B³ addresses and overcomes this obstacle. And you can see that after an intravenous injection of our antibody, the blood-brain barrier becomes open to a red fluorescent drug, which can now reach the brain, as you see on the left panel, whereas on the right panel, without the antibody, the drug remains inside the blood vessels and does not reach the brain. In practice, we inject this antibody intravenously at low doses, followed by injection of drugs. Here, a small molecular drug called cadaverine, and we can more than double over a period of 30 minutes the amount of drug delivered into the brain, as quantified on the left side, and you can see a visual on the right side. Again, the drug reaches the brain in the presence of the antibody, but not without. There are no effects on permeability of vessels and other parts of the body, and the effect is reversible. It lasts less than 8 hours. If we inject the drug 8 hours after our antibody, we no longer see the leak. And this is because the antibody is unstable and rapidly degraded, and this enables this transient on-demand blood-brain barrier opening. So we can deliver nanobodies. We can deliver bioactive molecules less than 70 kilodalton in size, and these include nanobodies that can be engineered against any brain target, growth factors such as BDNF that increase in the brain, but their plasma concentration remains the same. And this delivered BDNF is biologically active because it can phosphorylate cognate TrkB receptors in the brain. We can also enhance tumor cell killing by delivering chemotherapeutics that normally do not cross the blood-brain barrier, such as vincristine, as shown here, or doxorubicin. So we believe that our technology has a significant market potential for brain cancer, brain metastases, neurological diseases, and infections. And let me remind you that the global CNS drug delivery market size is estimated at $130 billion in 2022 and will grow even further over the years to come. We are competitive as our antibody technology is the only technology that is based on the transient interruption of a known mechanism required for maintenance of the blood-brain barrier that enables reversible, non-damaging, and brain-wide delivery of drugs, and actually it enables delivery of various drugs, and it can be injected intravenously. And this is the publication on the right that shows this from our lab. D²B³ is based on rigorous science. We developed the antibody against a target which is called ANG5B. This is an endothelial transmembrane receptor and guardian of the blood-brain barrier integrity. And we showed that this antibody blocks the binding of a ligand to the receptor, and this enables blood-brain barrier opening by changing the expression of permeability proteins on the endothelium. So I discovered the function of this pathway, and I'm a world authority in neurovascular research. The antibody we generated is a fully monoclonal antibody. We isolated it from a synthetic human antibody library by screening against the ANG5B extracellular domain. We have 10 hits, and we're testing 9 backup hits together with CROs that produce and test our recombinant antibodies. And our lead that I presented to you has very high affinity to both human and rat ANG5B. Therefore, it can be directly applied to humans and tested in preclinical rodent models in the lab. We don't expect any significant toxicity, as similar antibodies made against the ligand have shown good safety profiles in human studies. So going forward, we use the Blavatnik funding to have CROs reproduce our transient blood-brain barrier opening in their hands. Dose escalation studies are underway and tox studies as well. Backup antibodies are being tested, and in vivo proof of concept studies will be completed by the end of this year, focusing currently on chemotherapeutics delivery and their preclinical efficacy in brain cancer. We will branch out afterwards into brain metastasis and other indications. We are seeking partners for lead optimization and IND enabling studies, for which we estimate $5 to $8 million will be needed. Thank you very much for your attention, and here's our contact information should you be interested in contacting us.
Thanks, Jane. How about a round of applause for everybody? All right, thanks everybody for coming.
Got it. Oh, okay, very good. So like you get to like adapt to like the time zone when you went to okay, so you guys were the pitch did you sample the scene? So, okay, you want to go to invite myself to your dinner or hang out? I don't know, actually I can't remember which way I can go. I'm like asking your love.
We like
yeah, she's here. I don't know I'm a postdoc right now. Yes. Yes, I work with Pyle. Right here in your so I recognize your name here. I'm working on protein engineering of enzymes, serotex, Choukri Mamoun's infectious disease startup. We're doing small molecule stuff, so probably that's where I'll go. Element is Wuxi Biologics and the antibody business.
That's the thing.
So yeah, if we end up getting a chance to go dinner, I'd say like let's go to Melnick, and then time works out comment. I am not going to bio next year. We're not. How about first customers for any specific?
Yeah, so that's a good idea. You got to make your own. You just got the tape. So you have at least like a nice powerful but I feel like